going to be going over protein synthesis this morning. We're going to kind of break it apart into a couple of different pieces. We'll first hit, it's basically broken up into two different steps, and so we'll cover the first step this morning. Just kind of a refresher. If you look, there are the three different types of RNA. I think we've gone over these just briefly, but messenger RNA, this is a particular type of RNA, and its main function is to carry your genetic information to your ribosomes. It's going to be a copy of your DNA. Your transfer RNA does exactly what it says. It's going to transfer things. Specifically, it's going to transfer amino acids to your ribosomes so you can build your protein or something like that. So, and then your ribosomal RNA or your rRNA, this is going to make up your ribosomes. So it doesn't do too much else other than to just make up your ribosomes. All of these are still going to be single-stranded. They're all going to use that uracil instead of thymine, and they're going to use the ribose sugar. So it kind of shows you over here in the picture structurally how they're different just at first glance without looking at like the base sequences. So RNA is single-stranded, DNA is double-stranded. So. Okay, so the purpose of protein synthesis, if you break apart the words, protein is kind of is given, and then synthesis to make. So protein synthesis, your end result is to make a protein, or in this case, a polypeptide. So one more word that I want you to be familiar with is a word called codon. This is a series of 3 bases. We tend to refer to it as a triplet code, but it's just going to be 3 bases on your either DNA strand or your RNA strand. Usually they are going to be on your RNA strand is what we're going to reference. So later we'll talk about an anticodon, but for now, just understand that it's a sequence of 3 different bases in a row. So, like I said earlier, the process of protein synthesis is divided into 2 parts. The first part being transcription, the second part being translation. Transcription is essentially the copying of your DNA onto your messenger RNA and then moving it out to your ribosome. All of that's going to happen in the nucleus. Translation is then going to be translating the language of your DNA, or in this case your RNA, because it's the one that's the copy, into proteins. So you're translating your nitrogenous base sequences into a protein. Specifically, you're coding into coding for an amino acid. So this picture down here kind of shows the process of translation. So, but we'll get to that tomorrow. Transcription. So the first thing about this is, like I said on the last slide, this only occurs in your nucleus. It doesn't occur in the cytoplasm. It doesn't occur anywhere else. It's just in the nucleus. And the reason it's called transcription is because you're going to transcribe or copy your DNA or the information from your DNA onto messenger RNA. So this kind of shows you down here the picture. It kind of shows you that the DNA strand has been unzipped here, and then this messenger RNA strand has been built. It's basically the copy. Right here, this strand right here is essentially the same base for base as this right here, is this strand right here. The only difference is going to be, instead of having thymine, it's going to have uracil, but the sequence of the bases will be the exact same. Okay, so the very first step of transcription is that your DNA has to unwind. And just like with replication, it's not going to unwind the entire strand of DNA. It's just going to unwind little parts of it. And this is your template for your messenger RNA. Your proteins that are going to be building this messenger RNA are going to be looking for this specific series of nucleotides that are called the promoters. because that signals that this is where you're going to start transcription. So the specific protein that's going to be building the messenger RNA is called RNA polymerase, which is very fitting since in the last set of notes when we talked about replication, DNA polymerase built the DNA strand, so it's only fitting that RNA polymerase is going to build RNA. All right, so just like with replication, transcription can occur at various different points among the strand. So it doesn't just start at one end. It can start at multiple places. It can start at one end and start transcribing and then stay unwound and have a second RNA polymerase come in right after and start transcribing another copy of that messenger RNA, so you can have basically two copies of the same set of messenger RNA, and then you could have two of the exact same protein being built. So just for efficiency's sake. But okay. So after the DNA has unwound and after the RNA polymerase has found the promoters and it's started to transcribe, it's going to start building that messenger RNA strand. So it's going to read the DNA strand and it's going to build appropriately the responding or the matching or complementing, I guess would be the best word, complementing messenger RNA strand. So if your DNA strand reads ATGC, your messenger RNA is going to read UAGC because it's going to be complementing. So just like with DNA, it's going to build from the 3' to the 5' end, mostly for so that those proteins don't run into each other in the middle of building that RNA strand. Your bond between the DNA and the messenger RNA isn't very strong. So it's only going to be a couple of bases that are actually going to be physically attached to the DNA strand. So they're going to get lined up, they're going to get attached on the backbone with the phosphate groups and the ribose sugars, and then they're going to pretty much detach from the DNA strand. It's not like DNA replication where those bonds get put together and they stay together. These are supposed to be very temporary bonds holding the DNA and the messenger RNA together. So the last step, once the RNA polymerase has completed building the messenger RNA strand, that messenger RNA strand is completely released from that DNA molecule, and then the DNA molecule winds back up, curls back up, and it basically just waits until it's needed again to create more proteins. So, or specifically more messenger RNA strands. And then it'll leave the nucleus. So this is a picture kind of that shows you the transcription here. So here's your DNA strand. It's unzipped here. It's showing you that this messenger RNA is starting to be built. There, if you look, you can see the purines and the pyrimidines because they didn't really label this with A's and G's and things like that. But if you look, this strand right here is exactly the same as this strand right here. Oh, I guess, no, those are sugars and phosphates, I guess, right here. So, like I said, sorry, the only difference is that on the RNA, you're obviously not going to have the thymine. It's just going to be substituted for uracil. That's it. So, but that's basically transcription in a nutshell. It's pretty simple. However, in eukaryotes, there's one more step. There's some extra processing that has to go on before it's released out into the cytoplasm, the messenger RNA. So in eukaryotic cells, your DNA strand contains these two different things called introns and exons. These are groups of bases, are called introns, and then another group is called exons. Okay. Exons contain information for producing your specific protein. That's the information. Those are the bases that are actually going to be necessary to create the polypeptide chain here later. The intron's specific function we're not really sure about. All we know is that after the messenger RNA has been transcribed, it goes through this other processing, and these introns are actually cut out of your messenger RNA strand. So they don't actually leave the nucleus. So they're just going to get cut out. Things called spliceosomes, which are it's a protein made out of RNA, or made with RNA. That's going to do the cutting out of the introns. So it's basically going to cut out these introns. So a couple of bases in your messenger RNA sequence, and then it's going to cut out those introns and then paste these exons back together. So if I went back here, maybe, I don't know where I am. Well, now I'm typing. There we go. So let's say that these two bases right here, maybe everything's going crazy. If these two bases right here are the introns and these bases are exons, there is no rhyme or reason other than if these are the exons, these are the important bases needed to make. They're going to carry the information to make that protein. So messenger RNA is still going to copy it, but then what's going to happen is these, I guess it's down here, these two would probably get cut out, and then the rest would get pasted back together. So they would cut out the introns, and then this, if this was part of an exon, this would be pasted down to here, where if this was where the exon, the next exon started. So we would just basically get rid of this information right here. So that's basically what introns and exons are. You might run into this when you get to college. They might tell you a little about introns and exons. It's just another step of processing that only eukaryotes go through. Prokaryotes don't go through this. So they actually don't copy the introns as they're making the messenger RNA. They can self-splice, which almost seems weird because you would expect that out of a eukaryote cell, perhaps, than a prokaryotic cell. So, but this is basically transcription in a nutshell. So if you have any questions, just let me know. But this is pretty much where we're going to end today, and we'll pick up with the second part of protein synthesis tomorrow. So again, just let me know if you have any questions.